Chapter 10, Muscle Tissue. What we're going to cover today is the actual mechanism by which a contraction takes place. And just to review so we can become familiar with where we're going, let's take a look at what we talked about in the last lecture, which was the sarcomere. So this is the basic structural unit of a myofibril, and it is the contractile part of a myofibril, and it consists of thick and thin filaments arranged in a pattern so that the thin filaments are overlapping certain portions of the thick filaments, in fact overlapping where the heads are located, and completely surround, if you will, in this fashion, the thick filaments. We talked about the H zone, the A band, and the I band. We talked about the M line being the center of the sarcomere, and the Z disc being defining the edges of the sarcomere. So what we're going to do now is we're going to see how this sarcomere will shorten, the exact mechanism by which that takes place. So let's begin. And to do that, we will identify this as the contraction and relaxation of a skeletal muscle cell. So this is the contraction and relaxation of skeletal muscle cells. And the term that describes the mechanism in which this will take place is called the sliding filament mechanism. Okay. All right, a couple things to keep in mind as we describe this mechanism. Number one, there will be no change in the size or length of thick and thin filaments. The sarcomere will shorten. This sliding filament mechanism is composed of 4 phases. So, sliding filament mechanism is composed of 4 phases. 1. ATP hydrolysis. 2. Attachment of myosin heads to actin and formation of cross bridges. 3. Power stroke. And 4. Detachment of myosin from actin. So what we're about to describe then is this sliding filament mechanism, and we're going to call that, or we could describe that as the contraction cycle, because these four phases are going to repeat themselves over and over again as the muscle is contracting. So we'll identify this as the contraction cycle. So prior to the sliding filament mechanism being brought into play, we're going to have to release calcium. So how do we see this? Before the sliding mechanism, or I should say filament, mechanism, we are going to stimulate muscle cell by a motor neuron. This will be at the neuromuscular junction. Release of choline will cause, I got it right first time, a action potential of the muscle cell. The action potential will cause the sarcoplasmic reticulum to release calcium. Okay. So before the sliding filament mechanism can take place, these steps have to happen. We're going to stimulate the muscle cell with a motor neuron at the neuromuscular junction. That will cause the release of acetylcholine, which will cause an action potential in the muscle cell. The action potential will cause the sarcoplasmic reticulum to release calcium. Okay. Calcium binds to troponin, The troponin, tropomyosin, complex moves off and exposes the myosin binding site on actin. Now, the contraction cycle begins. So, let us take a look at let me grab these. Let's take a look at the details that is happening up here by way of two different pictures. So if you don't have these, you should go get them, and we'll talk about them, or we'll illustrate and use these as so there's one that looks like this, and you'll notice that there's no calcium being released at this point, so that's one drawing. And the other drawing is we see calcium being released in this next drawing. So we're going to use both of those to explain the activity and the actions of the action potential and the set of processes that happen before the contraction cycle. Okay. So let's begin by naming some structures. So this structure here is the sarcolemma. That is the cell membrane. This is my T-tubule. This is my terminal cistern. And then we have two structures here, which I will now highlight. So we have a structure here, and we have a structure here. Two different structures. So we're going to talk about those because that's the key to what we're going to do in terms of releasing the calcium. So we've got an action potential that comes down the T-tubule. So that's my action potential. But before that happens, the muscle is in a relaxed state. It's in this state that we see right here. And so what we're seeing here, and let me color those in. Calcium is being stored in the sarcoplasmic reticulum. So that's my calcium. And that's being stored in this sarcoplasmic reticulum. There's none or very little outside the sarcoplasmic reticulum. So the question is, how do we get that to be released? And the answer is these two proteins that you see here. So this one right here is a voltage-gated calcium channel. Voltage gated calcium channel. This one right here is called a calcium release channel. And what you're seeing is that the voltage-gated calcium channel is covering up this opening, which is the calcium release channel. So this is actually a channel in the membrane of the sarcoplasmic reticulum. And at this state right now, which is the relaxed state, the muscle is in the relaxation condition. What you're seeing now is that the voltage-gated calcium channel is covering up the opening of the calcium release channel. That's going to change with the action potential. So what's going to happen is the action potential comes down. It's being measured by the voltage-gated calcium channel. And when that channel, when this protein in the membrane in the sarcolemma of the muscle cell registers that action potential, it will move away and expose the opening of the calcium release channel. And so in the next image, we see this. So we have our action potential. And we see that the voltage-gated calcium channel has moved away from the calcium release channel. And that allows for sodium to leave the sarcoplasmic reticulum and bind to troponin, causing tropomyosin to roll off and expose the binding sites on actin. Right? In the previous drawing, you'll notice that there is no calcium binding, and the binding sites are covered over by tropomyosin. That's in the relaxed state. In the contracted state, we're going to get the binding of that calcium and the opening up of the calcium release channels, releasing calcium from the sarcoplasmic reticulum. That floods into the sarcomere, binding to troponin, causing tropomyosin to roll off the actin and expose the binding site on actin for the myosin heads, and we're going to form a cross bridge. So we've got the cross bridge formed because calcium is present. One last piece of information here, and that's this structure right there. This is a calcium pump, and it's constantly pumping calcium back into the sarcoplasmic reticulum. It never stops pumping calcium back into the sarcoplasmic. It's always running, never stops. So in the relaxed state, it removes all of the calcium. Well, not all, I shouldn't say that. A larger percentage of the calcium is removed from the inside of the cell and stored in the sarcoplasmic reticulum, because this is constantly working. Any calcium that comes along is immediately pumped into this structure, the sarcoplasmic reticulum, and stored there. So calcium levels are very, very low here in this relaxed state. But during contraction, we've stimulated that cell, we've opened up the barn door, calcium is leaving in large amounts, and that pump cannot keep up. So even though it's busy trying to put calcium back in, the amount of calcium that's escaping is greater than that pump is capable of dealing with. And so you get a buildup of calcium inside or around the sarcomere, as much as tenfold increase in calcium from the relaxed condition. Okay. So we've set the stage now for that cycle that we've described. And what I'm going to do next is I'm going to, in your notes, step by step, describe the contraction cycle. And that is very distracting, so I'm going to get rid of those. The contraction cycle. So maybe you hear the ice cream truck out there. I wish I could go get an ice cream, but I'm in the middle of a lecture. And maybe you hear the helicopter because we've got a demonstration going on here in Oakland. Okay, so let's start with the contraction cycle. And we'll simply put down in words what that cycle is, and then we'll use a drawing to illustrate that cycle. So the first step, number 1, ATP hydrolysis. And that includes the following steps. A, myosin head binds to ATP. So if you remember, the myosin head has a binding site for ATP, and ATP will bind there. Then ATP is going to react and become ADP, adenosine diphosphate, plus a phosphate group. The next step then is ADP, and the phosphate group is still attached to myosin head. What is this step is all about? This step is transferring energy from ATP into the myosin head. And the myosin head now will become energized. So basically it takes on a 90-degree orientation. So the myosin head is energized. Next step, number 2. Attachment of myosin head, gosh and golly, to actin to form cross bridge. So now the energized myosin head attaches to myosin binding site on actin. Okay. And we will release, oh, by the way, only one head binds to actin. Remember, a myosin molecule has two heads, but only one is going to be attached to the actin. The other one is still free. Next step then is we will release phosphate group. That will initiate the next phase, which is the power stroke. So now we're in the power stroke. So the release of phosphate group triggers power stroke. Myosin head rotates or swivels toward the M line, which is in the center, as you know, of the sarcomere. After the end of the power stroke, we will release ADP. The movement of the myosin head slides, and hence the term sliding filament mechanism. The thin filament, remember it's attached to the thin filament, past the thick filament towards the M line. So the power stroke, remember that the myosin head initially was at a 90-degree angle. And then, so this is energized. And then we get the power stroke. And what we're going to get now is the head at a more or less 45-degree angle. So it moves from here to there. Moves from a more or less 90-degree orientation to a 45. And because it's attached to actin, it drags the thin filament along with it. So this is my thick filament. Attached to it is the thin filament. And when this head swivels, it's dragging the thin filament with it. Where did it get the energy to move? From the hydrolysis of ATP. So when we broke off that phosphate group, the energy that was released was transferred to this head. It became energized. And then we released that energy with the power stroke. Number 4. Detachment. Of myosin head from actin. So at this point, the actin and myosin head still attached. And this is at the end of the power stroke. They're still connected to each other. So what's going to happen is we're going to, ATP will bind to myosin head and that will then cause the detachment detaches from actin. And then we start the whole thing over again. The cycle repeats itself over and over. So it takes 2 ATP molecules to complete the cycle. The first one, we're releasing the energy into the head. We're energizing the myosin head. And the second one has to come along to detach the myosin head from the thin filament. And then we add more energy to the system. So let's go ahead and draw, or not draw, well, let's go ahead and look at the process now by way of a set of pictures. So we've described it in our notes, and now we're going to go ahead and walk our way through this again using these images. So first step, myosin head is hydrolyzes ATP and becomes reoriented and energized. So we're going to get this combination of ADP and phosphate group. That head is now cocked. It's like a spring that's been pulled back and compressed. Okay. We have calcium present. That means that the actin molecule is exposed, and we can attach the heads to actin. The next step then would be the myosin heads bind to actin to form the cross bridge. And at this point, we're going to lose the phosphate group. And then we get the power stroke, the movement of the head from this position to, they're not really showing the position that well, but it doesn't matter. We're going to cock it and then release it. And when it's bound to and connected to the thin filament, that movement is dragging that filament with it. That is going to happen in the power stroke. Okay. So this is our power stroke right here. We get the movement. So we get binding and then movement. And then we are still connected. To disconnect, we have to bring in another ATP molecule, bind to it, and release that head. And then we go back to the cycle. So we energize the head. We connect to form a cross bridge. We get a power stroke. And then to release the cross bridge, we need another molecule of ATP. And then we'll energize it again. We'll go through this cycle over and over again. And what's happening is the movement of the sarcomere. The Z lines are moving towards the center of the sarcomere, and we're collapsing the sarcomere as we contract that system. Okay. Oakley Doakleys. So let's do our own drawing. Okay. Let's go just through and mark it up just as we see it here. Okay, so in the first step, this is step number 1. ATP is hydrolyzed. And we get ADP and a phosphate group. We get this 90-degree orientation. Calcium is present because we have the exposed binding sites on actin for the myosin head. But that head cannot bind unless we have this configuration. It's energized and we have the combination of ADP and phosphate. Then we get cross bridges. So step number 2, cross bridges formed. And we get the release of the phosphate group. So we still have ADP and phosphate here. We form the cross bridge, we lose phosphate, and we still have ADP connected. And now we get a movement, a sliding. So this is our power stroke. And we get this movement towards the M line. Then we release the ADP, and we get ATP to come in. ATP binds, and we get release in the third step. So this is number 2, this is number 3, number 4 is detachment. Okay, that is the sliding filament mechanism, and we're going to get, at that point then, we're going to have contraction in the muscle. Couple things to keep in mind. This is a all or nothing proposition. All or nothing proposition. If we have calcium, if we have ATP, this will continue. There is no, how should I put it? There's nothing between 0 and 60 miles an hour. It's either going to be complete cycle movement of the and sliding of the filament or nothing at all. We cannot calibrate this. We cannot make this go slower or faster. It's just going to happen in a continuous cycle. So that brings up a question which we're going to address in the next lecture. How do we, because this is an all or nothing proposition, how do we exert different amounts of force using our muscles? Because obviously we don't use all of our muscles to do every kind of job or movement. So how do we calibrate the system? If this is an all or nothing system here that we're describing, how is it that we can apply less force or more force depending on what it is that we want to do? And that will be answered in the next session. Okay. Let me talk a little bit about calcium. So again, if we have an increase in calcium, we have a muscle contraction. If we have a decrease in calcium, we have a relaxed muscle. No contraction. So calcium is the on-off switch for a muscle contraction. Okay. Calcium is stored inside the sarcoplasmic reticulum. We can get very high concentrations of calcium inside the sarcoplasmic reticulum. So how does that calcium get into the sarcoplasmic reticulum? We have calcium active transport pumps that move calcium from the sarcoplasm to the inside of the sarcoplasmic reticulum. So inside the sarcoplasmic reticulum, there is a molecule called calsequestrin. This is a protein. So originally, the sarcoplasmic reticulum was the rough endoplasmic reticulum, and it was building a protein as the cell was developing to become a fully mature skeletal muscle cell. That protein is calsequestrin, and what that does is it binds to calcium inside the sarcoplasmic reticulum. Why do we have this molecule? Because we have a problem. Consider. So the concentration inside the sarcoplasmic reticulum of calcium can be 10,000 times greater than the sarcoplasm. That's a lot. So if you have a space and you have a lot of calcium in that space, and we have very little out here, and we know that calcium is not going to be able to cross that membrane. That's a selectively permeable membrane. What's going to happen? Water is going to move in to that space. How do we prevent that from happening? What we do is we put a protein in there, and I'm just going to choose a random color here. I have no idea how this protein looks. We're going to put a protein in there, and that protein is going to bind to and soak up like a sponge all the calcium. So now the calcium that's bound to the protein is out of solution, and we have we don't have that problem of water moving across the membrane because we don't have that huge concentration of calcium inside the cell because it's all attached to this protein called calsequestrin, and that solves our problem for us. All right. So that's it for this presentation. And in the next presentation, we're going to talk about developing tension in muscles. How do you calibrate force using the muscles? Because we've all described here a all or nothing proposition. So we need to find a way that we can apply less or more force depending on what kind of job we want to do with our muscles. And that's what we'll cover in the next lecture. So I'll see you there.